Hello, my name is Ethan Jackson. Today I will be discussing color blindness, or color deficiency, a condition that exists within my family. Color blindness, also called color deficiency, occurs when one has the inability to distinguish certain colors. It is oftenly inherited versus a caused condition. In one's retina, there are rods and cones. Rods detect light and dark, where cones detect color. This deficiency exists when one's color receptor cones are either absent, defective, or if they process a different color than a normal person would expect. It's estimated that 300 million people in the world suffer from this condition, 1 in 12 being men and 1 in 200 being women. This makes 95% of the colorblind population men, and 98% of the entire population suffer from red-green color blindness. Sex-linked genes were first discovered by Thomas Morgan in 1910 when he found a disproportionate percentage of white-eyed male fruit flies among his population. This topic was discussed in Chapter 4. Traits determined by the X chromosomes are termed X-linked, and those by the Y are Y-linked. X-linked traits can either be dominant or recessive. Color blindness is an X-linked recessive gene. We can see here that it requires both affected alleles on the X chromosome to have an affected female, and while the male only possesses one X chromosome, he will only need the one affected allele. Looking at these Punnett squares, one can estimate whether or not their offspring will be affected, non-affected, or carriers if they know their own genotypes. Estimated genotypes leads us into Chapter 6, Pedigree Analysis. A pedigree is a pictorial representation of a family history, or a family tree outlining inheritance of characteristics. A pedigree allows us to determine modes of inheritance of a certain trait running through a family, as one can see the genotypes of certain affected individuals if known. Looking at this pedigree of my own personal family, we can study how color blindness has affected my family. Non-shaded squares and circles are unaffected males and females, completely shaded are affected individuals, and half-shaded are carriers. One can see there are no half-shaded squares in the pedigree, as color blindness, being an X-linked trait, cannot produce carriers in men. My great-grandfather was unaffected, yet because my great-grandmother was a carrier, they were able to have an affected son, my grandpa, who then, with my carrier grandma, had my affected father. And even though my mother was completely unaffected, my sister must be a carrier, as she must take the affected X chromosome from my father. When studying the mutation of color blindness, we can find it is an indel mutation, which we studied in Chapter 18. These are insertions or deletions of base pairs within a sequence of genetic code. Unless the indel is divisible by 3, it is a frameshift mutation and can have a drastic change on the codons, changing most, if not all, of the amino acids following the mutation. The X chromosome has L and M opsins, which lie in the head-to-tail array on the distal tip of the long arm of the X chromosome. This region is termed XQ28. Deletions in this array are the main cause of the red-green color blindness we see in 98% of the colorblind population today. Thank you, that is all I have today about color blindness, but using pedigrees you can map out your family history to follow the inheritance path of color blindness or other traits to determine the chance you'll pass them down to your own offspring.